Right.
Oh dear. Okay. So clearly, you guys aren't coming just because you love me. Okay, here we go. So we're starting DNA damage and repair. Is that what we want? Yeah, DNA damage and repair. And that will be the last topic we'll cover, and then you'll have to leave me and move on to bigger and better. I mean, you can feel free to stay and hang out with me. Yeah,
Approximately when will we be getting our exam grades back?
never. You'll get them back as soon as they are graded.
Sounds good.
I will always get them back to you 1 week after the last exam is taken. So the last exam was taken today. You'll get them back at the absolute latest Friday, next Friday. Because I'm guessing you're trying to figure out if you want to take the final. So don't worry about it. I like to, hold it off long enough so that you'll be forced to come to class and not just fail on class altogether. Okay. The final exam is May 20th. Gene expression project is due next week. I had nightmares about that last time.
What?
Just so you know,
Nightmares?
I had nightmares. There's too much going on in my life and I'm losing my mind. But that's another story. Yeah, there was one week left of class and we needed to take the final and the gene expression project was due at the same time, and I had to try to fit them both into the same recitation section. And I had no rubric prepared for grading, and then there was like an observation, so the faculty were watching. Then there was a fire alarm, and the people had to yell to leave. It was bad news. So that's not going to happen. I was wearing pants, at least. Okay. If you need help, I'm getting lots of emails from people about who are needing help with their project. I feel like if you're at the point where you're like, I found this new piece of data and it doesn't fit in, what do I do? Do I have to start over again? No. Power through, because just because you find one paper that doesn't agree with your hypothesis doesn't mean it's wrong, because that paper might be wrong. And it's a proposal, and it's not real life. Well, it is real life. But it's a proposal, so you just go with it, and if you come through logically, I'm not going to look for every single proposal to check all the literature out there to make sure there's nothing against what you're saying. I mean, that's crazy. I don't have that kind of time in my life. I'm going to look for logic and an ability to form a hypothesis and 3 aims and talk about that. If you are at the point where you're like, oh my god, my aims are horrible and my group is falling apart and I'm crying every night, make an appointment to see me, because in email I can only do so much. I can look quickly at something and say that looks pretty good, but I'm not really sure. Because you now are the experts in this. I can, especially I got a really nice email with a great project. But I can't tell you definitively if it's perfect, because it's a whole pathway I have to sit and think and analyze to decide whether or not it's perfect. So if you can show me you have logical conclusions and logical steps to take, you're good, okay? But it's really hard to just respond to long emails, long panicked emails. So try to make an appointment to come see me. I know we're running out of time for that, but I'm just saying, let's try to work that out. Okay, so far we talked about translation. Specifically, we spoke about the mechanism of translation. We talked about the how the amino acids are added to tRNAs and how that is done in a correct way and how important that is for making sure the right amino acid is added to the growing polypeptide. And we talked about translation initiation, elongation, and termination. That's pretty much what we did the last 2 days. Now we're on to DNA damage and repair. Okay, so before we start going through this, you should acknowledge the fact that there are many diseases associated with defects in DNA damage and DNA repair, which makes sense. We're constantly being attacked by environmental insults to our DNA or metabolic insults to our DNA. And so having multiple repair mechanisms is critical for survival, really. Critical for not getting a whole lot of cancer, right? So here are 1, 2, 3, 4, 5, 6, 7, 8, 9 different diseases or syndromes that are associated with different processes or enzymes involved in DNA repair that are defective. Everything from the whole mismatch repair system to just DNA ligase 1, and most of them you can see have to do with UV sensitivity and cancer, genome instability, which makes sense, right? You need to acquire multiple mutations to get cancer, so if your repair mechanisms aren't working, you're going to acquire those mutations a lot faster. Okay, so this is me getting you to care about this. Do you care? If you're here, I'm assuming you care, right? Because everyone who really doesn't care and just wants to get through and get on to the next step in their life, they're not here. Okay. Sorry, guys. I know you love me. I'm talking to my computer. Okay, so left unrepaired, DNA damage leads to base deletion or substitution in the daughter DNA molecule after DNA replication. That's one of the things that happens. Also, it could just completely stall DNA replication, which could result in cell cycle arrest or apoptosis. And so when you think about DNA damage, you really need to be thinking about, well, how is this going to affect replication? So if you have a C that's deaminated into a U, right? Then when you have DNA replication, you're going to get this parental strand is going to be fine. So these are going to be fine. But here, you're going to get base pairing with an A. So now you have new sequence showing up. Okay? And this is how the mutation arises. For example, if you get depurination, which means removal of a whole purine, then when you have DNA replication, this side will be fine, but this parental strand is missing a whole base, and so you'll get deletion of a base. So that's one way that deletions arise. Okay? How do we feel about this? Yeah, okay. So you can get spontaneous DNA damage from many ways. You can get, there could be oxidative damage from reactive metabolites and environmental chemicals. Can get just plain old hydrolytic attack and uncontrolled methylation, not the methylation we want. Methylation on other places other than 5-methylcytosine. For example, according to the textbook, about 500 purines are depurinated and about 100 cytosines are deaminated per cell every day under normal conditions. This is normal. This isn't like someone who's a heavy smoker, or someone who's working in a toxic factory, or someone who's undergoing chemotherapy. This is normal. So it's kind of amazing that we're here, but that's, of course, because we have all these systems to repair DNA. Okay, any thoughts? This is kind of mind-blowing, right? Oh, then again, if you think about the size of our genome, it's not that much, right? But still, it's significant. Yeah.
So what happens in depurination after the purine's gone?
Well, we're going to go through a whole bunch of mechanisms for repair. Yeah. So let's first talk about damage, and then we'll talk about repair. Okay. So yes, you can have depurination. So that's where you have a purine group that is lost, and then you're stuck with an abasic site, right? So there's no base there. Or you can have deamination, where your cytosine is deaminated, so there's your amide group right there that goes away, and you'll end up with oxygen there, and then that's a uracil. Should uracil be in DNA? No. Yeah.
Is that figure, the 500 purines and the 100 cytosines, is that expected that it's always been the case, or is that grown over time?
Oh, so you're wondering if environmental factors like the Industrial Revolution and whatnot have
Right.
affected this. I have no idea. Because,
Even increased UV.
well, UV causes thymidine dimers mostly. So I guess we need to think about what's causing all of this, and I think a lot of things, there's normal metabolites in your body that cause this. But yeah, I'm sure things have changed over time. I don't know, but then you can argue, so that goes into a whole conversation about the incidence of cancer over time, and people say the incidence of cancer is increasing. Well, really, is it? Or is it just that we're living longer and our detection is better? One would guess it probably is truly increasing. So, nice food for thought. UV radiation from the sun. So I like this. You know, you're going out for a run in the nice summer sun, and really what you're doing is you're getting UV irradiated, and unless you put sunscreen on it, then you're fine. I'm horrible about who puts sunscreen on. I'm horrible about it. Okay, it's all the fair people in the room are putting sunscreen on, and all the people who are dark skinned or tan. I turn like brown in the summer, and I never put sunscreen on. I'm gonna end up with cancer, I'm sure. Sunscreen on. But yeah. So this is me running because I haven't run all year because my knee's been injured, so I look like that, and I've turned into a man. And the sun is irradiating my un-sunscreened skin and creating these thymidine dimers. And so it's actually covalent linkage between two adjacent pyrimidines in the DNA. And is that good when you have to replicate your DNA? No, it's not good, right? So that's one form of DNA damage. And the other thing is it messes with the structure of the DNA. Then there's alkylating agents, and this is my favorite to talk about at Towson University because of the excess of bacon in our diets here at Towson University. Who loves the fact that there's bacon on everything? Okay, well, who hates it? Thank you. It makes me crazy. Can't get a turkey sandwich like pre-made without bacon infiltrating it. So alkylating agents are byproducts of metabolism that add methyl or ethyl groups to bases. So here's a nice happy guanine, and an alkylating agent might add a methyl group here, and then that'll mess with base pairing, so your methylguanine will base pair with T instead of C. And then on a second round of replication, the T pairs with A, and you change your sequence. So it disrupts the normal base pairing interactions, and it can also cause interstrand cross-links, which will block strand separation. When do you need to separate your strands of DNA? Replication, right? So that's not good. And so an example of an alkylating agent are nitrosamines and cured meats. And in addition to cured meats, I'm not just going to give bacon a hard time. It's also other meats and fish and beer. And it's also just a byproduct. I know being a man in America basically is going to cause you to have lots of nitrosamines. I just made a judgment that I shouldn't have made, but that's okay. So the example, I know men like bacon and beer. So you'll have your so meat contains amines, and sodium nitrate is added in the curing process, and then when you cook your bacon at a high heat, it causes formation of these nitrosamines. But the good news is, I read a little bit about this because it was so interesting to me. So there used to be a whole, there was this balance. You want these nitrates in because, or sodium nitrate because you want to prevent botulism because it's a preservative, but you don't want to cause cancer and DNA damage because it is associated with cancer. So there was like the arguments between the cancer people and the food-borne illness people. So they decrease levels of sodium nitrate in foods, and you can add in things like ascorbic acid that are going to prevent formation of nitrosamine. So it's not as horrible as it was, but still, just normal metabolism of meat can cause formation of nitrosamine. So we're kind of all screwed. Unless you're a vegetarian and you drink beer. And in that case, you're still screwed. Unless you're a straight edge vegan, then you're okay. Okay, were you raising your hand?
No.
Or you were really happy that you don't eat meat?
They have that drink beer, so.
Oh, you're in trouble. Okay. So that's my story about bacon. Okay, so that's enough about alkylating agents. Intercalating agents are aromatic or flat hydrophobic molecules that become inserted between the stacked bases in the DNA double helix. Here's ethidium bromide. Who's used ethidium bromide?
Yes.
Yes. Who's dumped it on themselves by mistake? Okay, good. Who's drank it? Who put their ethidium bromide into their hot agros and watched it?
Watch them do it.
You don't ever really want, so you like heat up your agros to melt it when you're going to pour your DNA gel, and then you often will either add ethidium bromide directly, or some people like to soak their gel later in this massive vat of ethidium bromide, which I think is horrible, but everyone prefers that here. And if you put it into your hot agros, and it's steaming, and you're standing there looking at it, breathing in the I always tell people, step away, let it cool until it stops steaming, then put in your ethidium bromide. Anyways, ethidium bromide is used in the lab for staining DNA. You can see why it stains it so well, because it has an orange color, and it slides neatly in between all your bases. It intercalates. And this is definitely not good in your DNA, but it's fantastic for visualizing DNA on a gel. People use CyberSafe here also. I don't know, what are you guys using in the 410 lab when you run a DNA gel? It's ethidium bromide? People use CyberSafe because it's cyber green based, it's supposed to be safer, but I think if you're intercalating anything, if you're staining DNA, how could that be good for you, right? If it's something that binds to DNA and it gets in you, how's that good for you? But anyways, so that's the story with intercalating agents. So they extend and partly unwind the DNA helix when they slide in here, and it could result in insertions and deletions upon replication. So notice usually the poor outcome is after replication. Then there's ionizing radiation, which most of us have had because we've been x-rayed, right? And it causes single or double strand breaks. Not good, but that's why they cover you with the lead apron. Huh?
And then shoot x-rays at your
And
head.
then shoot x-rays at your head. So I was trying to understand how it caused single double-stranded breaks because they just tell you that and you don't know how. And so it has to do with the energy of the x-rays. And they can actually liberate an electron out of your DNA, which is what causes the breaks. Okay, so damage is occurring pretty frequently. And you guys all believe that. We're eating bacon, we're getting x-rayed, we're going running in the sun. Right? Lots of opportunities for damage, whether you're a smoker or not, right? You might think, oh, you shouldn't smoke, you're going to get cancer. Well, you're kind of going to get cancer anyways if you live to be like 85 or 90. So I'm not endorsing smoking, but I'm just saying. So why and how has our species and many others survived? Yes?
A really good repair mechanism for DNA damage.
A really good repair mechanism, or many good repair mechanisms for DNA damage, right? So we've evolved many repair mechanisms for DNA damage. And then the other thing to think about is our DNA is nicely packaged in a double-stranded conformation rather than single-stranded. That doesn't protect it from damage, but it provides a template for repair, right? So that's nice too. So I was So storage of genetic information in double-strand DNA provides a template for repair, and we have many DNA repair mechanisms. And I like that picture. It's from like the DNA damage working group at NIH. Look, see, it's like the guys, like the workers fixing the DNA. Okay, fine. Let's get into the end of the semester. I don't like blank slides. Okay, so when you start thinking about DNA repair, there's two major mechanisms. One is direct reversal, and I'm not going to talk too much about that. There are specific enzymes that recognize damage and fix it. So one is photolysis, that use energy from near UV light to split pyrimidine dimers. So it's reversing the pyrimidine dimers by splitting it. And then there are methyltransferases, different methyltransferases that recognize methylguanine and methylated backbones and repair. And then there's the other category, which is excision followed by resynthesis. So you're cutting out what's bad and resynthesizing using a DNA polymerase. Okay? You having fun? Okay, so here's an example of reversal by a methyltransferase. And this is in E. coli. ADA, I say ADA, you might say ADA, I don't know. It's an alkyl transferase, and it removes methyl groups from DNA. So here's your methylated DNA, and it activates expression of itself. So ADA, the enzyme, comes along, and it actually transfers the methyl group from the DNA to itself. That's why it's a transferase, but it's methylating itself. So it's removing the methyl group from the damaged DNA and adding it to itself. So it's a single-use enzyme. And it also, once it's methylated, this is very cool. Once it's methylated, it activates expression of its own gene and a glycosylase gene. Very cool, right? So it senses the DNA damage, it removes the methyl group, repairs it, and since it's a single-use enzyme, it's helpful that it upregulates its own expression. So you could guess that this has a DNA binding site as well, a DNA binding domain as well as a methyltransferase. Right? What do you guys think about that? Very cool, huh? No? Yes? Okay. People are nodding. So let's go back a minute. Okay, so we're talking about removal of things that should happen. Well, there's also errors that can happen during replication, right? We talked about DNA replication and this fantastic characteristic of DNA polymerase called exonuclease activity that removes incorrectly added bases. But sometimes some errors get past that, and there's another mechanism to repair them. And so this is called strand-directed mismatch repair. Strand-directed meaning it's using the template, the correct strand as a template for repairing a mismatch. Okay, so this is in bacteria. There's a similar mechanism in eukaryotic cells. So after replication, post-replication mismatch repair will repair mismatched bases missed by the DNA polymerase exonuclease activity. Right, so it's good. If something as important as our genome, we want to have multiple mechanisms to make sure it doesn't change too much. So first you have to recognize the mismatch, and you can recognize it because the structure will be changed if you have a mismatch. Normal base pairing should have the right structure, whereas you're going to have a bulge when you have a mismatch. Okay? And a protein called MUT-S will recognize this effect on the structure, and it will bind here, and it's interacting with the sliding clamp, so it's falling close behind during replication. And then this MUT-L protein will scan and direct a nick in the DNA. And, well, before I go through that, we need to somehow distinguish between the parental strand and the newly replicated strand, right? Because you want to know what to repair. So who remembers how that's distinguished?
With bacteria,
The bacteria.
it's hemimethylated.
Right, hemimethylated. Remember, bacteria likes to methylate its DNA, and there's a little bit of a time lag. So after replication, only your parental strand is going to be methylated, and the newly replicated strand won't. Good. So the new strand is distinguished from the parental, and then Mut-L scans and finds a nick in the strand, and then helicase will be recruited to displace it, and then exonucleases will degrade basically everything from where Mut-L is all the way to Mut-S, so remove that whole stretch of DNA, and then the DNA is resynthesized and ligated. So DNA polymerase and a DNA ligase will repair the DNA. Right, and so it's Mut-H that nicks the unmethylated GATC sequence somewhere down the road, and that's what determines the length that's going to be degraded. Okay, so recognize damage by change in DNA shape, nick it, degrade from damage until the nick, and then repair. Any thoughts, any questions? Good. Okay, so this also exists in human cells. The only difference is the way the template strand is recognized. Obviously, the methylation is not going to work for us to recognize a template strand. So what it actually does is it looks for nicks that haven't been sealed yet by DNA ligase. There are a lot of those that are in the ligand strand. Remember, you have the primer removal, and you've got the DNA polymerase that fills in, and then there's the gap that's sealed by ligase. So, defects in the system are associated with a cancer, a hereditary colon cancer, and that's because if it can't repair mismatches resulting from replication, you're going to really rapidly accumulate mutations, which, remember, lots of mutations that can't be repaired, cancer. Okay? Okay, so mismatch repair can also correct insertions and deletions that come from slipped strand replication. So here you'll have a situation where you're replicating and something will happen. Well, there'll be a secondary structure that might form in the DNA during replication. Now you would hope that your single strand binding proteins might prevent this, but sometimes these secondary structures can form, and that might result in skipping a number of bases, and that will result in a deletion or an insertion in the daughter strand. So here in this example, the deletion is generated from this hairpin that's formed from a repetitive sequence in the template strand. So remember, a repetitive sequence is going to be more likely to bind to itself. Okay, how's it going? Okay, so now let's go into excision repair. There's two kinds of excision repairs: base excision repair and nucleotide excision repair. And the only significant difference is in one you're just removing a base, and the other you're removing a whole bunch of bases. And so I guess by calling it nucleotide excision repair can become confusing. But just remember, base means base removal, nucleotide excision repair means a whole bunch of bases are being removed. And so the way it works is you might find there might be a deaminated C, so it's been deaminated and now it's a U, and a uracil DNA glycosylase, so an enzyme that's going to recognize uracils in DNA, right? That's handy. Is it handy to recognize uracils in DNA? Would it be handy to recognize uracils in RNA? No, because they're everywhere, right? And not a sign of much. Okay, making sure we're awake. Everyone awake? Yeah. Okay. Uracil DNA glycosylase is going to remove that base, And so now you've got a DNA helix with a missing base. So it's an A basic site. A basic, no base. And then a endonuclease and a phosphodiesterase are going to remove the sugar phosphate backbone. And then DNA polymerase comes in, repairs, and ligase seals the next. Okay? So it's really just remove the thing that's wrong and repair it. Right? Pretty simple. You can make it complicated, but it's pretty simple. Here, this is an example how you might repair pyrimidine dimer. Now we talked about photolinases. That's one way to split a pyrimidine dimer. But you could also sometimes have a pyrimidine dimer that isn't repaired that way for whatever reason, and you would repair it through nucleotide excision repair. And here, a nuclease would cleave on either side of the damaged base. The damaged bases are right here, and remove that whole section of DNA. And then again, DNA polymerase is going to come in and resynthesize, and ligase is going to seal it. Okay? So, base excision repair, single base removed, nucleotide excision repair, a short stretch of bases are removed. Any questions? I feel like I'm kind of just going on and on here. Now, does anyone want to see me do a dance? I don't have any dances today. Maybe Monday. Okay. This is another way to look at it. So you can have damaged DNA bases and A basic sites. Those are both repaired by base excision repair and resynthesis. So, and it's just what I told you before. You've got a base, and then it's removed by glycosylase, and then you have removal of the sugar backbone, and then you have repair by DNA polymerase and ligase. I don't know why I have that in there twice. So, damaged bases are removed by DNA glycosylase, and this creates a basic site. So here would be your damaged base. Here you have it changed in some way, maybe methylated, and DNA glycosylate is going to flip it out and remove it. So nucleotide excision repair, here enzymes remove short stretches of DNA, and again, you get recognition by these bulky lesions. So if you've got UV radiation or a chemical that's distorted the shape of the DNA, that's how nucleotide excision repair enzymes are going to be recruited to the site of damage. And it's usually about 10 to 30 base pair stretch that's removed. And it's filled by a repaired DNA polymerase. And so in this example, this is from, I believe, E. coli. So you'll have UVR proteins, and again, they're not very, they're kind of boring in how they name them, right? A, B, C, D. So UVR A and B, they're scanning for DNA damage. UVR B opens the DNA, and then UVR C nicks the 3' and 5' side of the lesion. So you have it open, then you have nicking, and then you have UVR D and DNA polymerase 1 release the oligo, and then it's repaired by resynthesis. So the nucleotide excision repair, nucleotide excision repair machinery in different organisms. There's a lot of it. It's different. They have many different proteins with different names but same function. So it's highly conserved, which would make sense, right? It would make sense that nucleotide excision repair, or just DNA repair in general, is conserved in bacteria and in eukaryotes. So the E. coli ones are these UVR ones. Yeast is usually RAD proteins. And then in humans, they tend to be named based on the diseases they cause. So there's a lot of XP going on, and that's from xeroderma pigmentosum, which is a syndrome that comes from defects in DNA damage repair? Okay, I think we are gonna stop early today. So those who want to eat something before the exam can do that. And we will finish, is next week the last week of classes? And then it's a Monday, okay, so we will get through this all by then.
So, I don't know if you remember, but a couple weeks ago, we were talking about slicing audience.